All right, let's dive right in. Today we're talking about the code behind the code of life itself. We're going to look at the two most foundational file formats in all of bioinformatics, FASTA and FASTQ. These are basically the text files that take the raw, chaotic output from a DNA sequencer and turn it into data we can actually understand and work with. Let's get into it.
So, picture this. You have these incredible sequencing machines just churning out mountains of genetic information. I mean, a single run can produce terabytes of data. That creates a massive question, right? How do you organize all of that? How is every single little strand of DNA or RNA captured and stored in a way that's even remotely usable?
Well, the first and maybe the simplest answer is the FASTA format. You can think of it as the original, no-frills solution. It's a plain text format for storing a sequence, whether it's DNA, RNA, or even protein. And its real beauty is in its simplicity. Honestly, it's not much more complicated than the digital version of just jotting down a sequence on a piece of paper.
The structure itself is incredibly straightforward. Every single record in a FASTA file has just two parts. First, there's a header line that works like a label. It always starts with a greater than symbol, and it gives you a description of the sequence. Then, right after that, you get the sequence itself, the raw string of letters, which can either be on one really long line or broken up across several lines.
Now, if you're coming from a database world, your brain is probably already asking questions. You're looking at that header line and thinking, okay, is that a strictly defined field? Is it something like a unique primary key that has to follow a certain set of rules to make sure every record is perfectly identifiable?
And that's exactly where we hit the biggest pitfall of FASTA. The answer is a hard no. That header is basically a free-for-all text field. There's no standardized format, which means you get wildly inconsistent naming from one database to another, from one lab to another. You combine that with the fact that the sequences can be split across any number of lines, and trying to reliably parse these files becomes a real headache.
Okay, so FASTA gives us the sequence. Great, but that leads to an even more critical question. When a sequencer reads a base, let's say a G, how confident are we that it's actually a G and not just a machine error? How do we store the accuracy of the data itself? Well, FASTA has no answer for this at all.
And this is the exact problem that the FASTQ format was designed to solve. It's the next step in the evolution. FASTQ takes that simple sequence from FASTA and it adds this crucial second layer of information, a quality score for every single base, telling you precisely how confident the machine was in that specific letter.
When you see them side by side, the difference in structure is immediately obvious. A FASTA record has 2 lines. A FASTQ record has 4. Line 1 is the identifier, but this time it starts with an @ symbol. Line 2 is the sequence, just like before. Line 3 is always just a plus symbol. It's basically a separator. And then there's line 4, that string of characters. That's where the quality scores are hidden. 1 character for each base in the sequence on line 2.
Right, that 4th line. It looks a bit like someone just smashed their keyboard, doesn't it? What on earth could those characters possibly mean? They look random, but each one is a precise, coded piece of information. So how do we decode it?
It's actually a pretty clever encoding system. Here's how it works. You take a character from that quality string, let's use a capital J as an example. You find its numerical value in the ASCII table. For J, that's 74. Then you subtract a standard offset number, which for modern files is 33. So 74 minus 33 equals 41. That final number is the PHRD quality score, a direct measure of accuracy. The higher the score, the better the quality.
Now, I should point out that in the early days of sequencing, there were a few different encoding standards floating around, each with its own offset value. It caused a lot of confusion. Thankfully, the field is now pretty much completely standardized on the Sanger format with its offset of 33. The crucial thing to remember is that you have to know which encoding your data uses to interpret those quality scores correctly. So this brings us back to a really practical challenge. For anyone who's used to the rigid, predictable structure of a SQL database table, these formats can feel a little loose, a bit chaotic. So how do you do a really simple task, like just counting how many sequences are in a massive file without having to write a custom, error-prone parser?
This quote just gets right to the heart of the problem. The ad hoc nature of these formats is both their greatest strength and their greatest weakness. Their flexibility is what makes them so universal, but it also means you can't just assume a clean, predictable structure. You need robust methods to handle all the potential inconsistencies.
And here's a beautifully simple and powerful way to do it. Instead of writing a complex script, you can use a standard command line tool like grep. To count FASTA records, you just count the lines that start with the greater than symbol. For FASTQ, you count the lines that start with @. This simple one-liner completely bypasses all the weirdness about line wrapping and gives you a reliable count in seconds. It's a perfect example of using the right tool for the job.
So, we've seen this evolution, from FASTA's simple sequence to FASTQ's much richer, quality-aware data. Both are text-based, incredibly powerful, and absolutely fundamental to biology. But they're also full of these tricky little pitfalls. And it all leads to a bigger question about the day-to-day work in bioinformatics. If the foundational data of modern biology lives in such flexible, almost artisanal formats, where does the task of just cleaning up the data end? And where does the actual process of scientific discovery finally begin?
And that's our breakdown of FASTA and FASTQ. If you found this useful for understanding the data formats that really underpin all of genomics, please hit that like button, share it with a colleague, and subscribe for more explainers that get into the nitty-gritty. Thanks for tuning in.